This is a presentation of the University of Wisconsin Parkside.
Good evening. On behalf of the Science Night Collective, I'm very pleased to welcome you here tonight to Science Night at the University of Wisconsin Parkside. Tonight's speakers are Drs. Brian Lewis and Paul Mahosby. Dr. Lewis is the Assistant Dean in the College of Natural and Health Sciences and Director of the Center for Health Sciences. He studied chemistry and biology at Ripon College, got his PhD at UW-Madison, and is widely known for his energetic lecture style, which I hope we'll see some of tonight. Dr. Lewis's duties here at the university do not require him to lecture very often, but he really likes to do it and goes out of his way, even when it's not strictly required. He volunteers for various teaching activities. Dr. Mahosby, Dr. Paul Mahosby, is professor of physics. He got his PhD in materials science and engineering at the University of California, Berkeley, taught at various institutions for 10 years, and has been at Parkside since 1986. He, too, is well renowned for his lecturing ability. In addition to winning the Research Award, he is a 3-time winner, and he may be the only 3-time winner of the Stella Gray Teaching Award here at UW Parkside. It's a particular pleasure to welcome Dr. Mahosby here because he, in fact, is the founder of Science Night. Over 10 years ago, inspired by the first modern philosopher, René Descartes, who believed that science and mathematics could explain and predict events in the physical world, Dr. Mahosby founded this series in order to bring science to the general public. Tonight, Drs. Lewis and Mahosby will be speaking about physics and the human body.
Well, thank you, Dr. Mayer, for the wonderful introductions. Thank you to our audience, students, community members, faculty, staff, friends. Just thank you all for coming out tonight. I know it means a lot to me personally to have you guys out in the audience. I know it means a lot to Dr. Mahosby as well. So hopefully we're going to get some knowledge today. Hopefully we're going to have some fun along the way, too. So sit back, enjoy, and feel free to ask questions at any time. So I think we've already kind of covered the general introduction. So we're going to be talking about physics in the human body. And one of the things that we're going to be talking about essentially is how does the body work? Now, when Dr. Mahosby and I sat down and started to think about what are we going to do tonight, we came up with a list of about 50 different things. So I hope you're strapped in. Okay. There are a lot of different physiological functions and anatomical functions that we thought we could cover and that would be able to relate to the field of physics and help explain some of these concepts and show how physics is real, how it's applicable, and how it's really fascinating and interesting, and also integral to life. Okay, you need physics in order to function. So when you think about science as a general field, the idea of splitting science into separate subdisciplines is a relatively new concept. Science used to be taught where it was biology, chemistry, physics, and mathematics as one subject area, one discipline area. That might have actually happened for you if you think back to like 7th grade. Okay, where it was just science. Okay, so when you're talking specifically about all these fields, they are clearly all interrelated, and you really can't explain one without the other. And one of the pure bases for this is physics. In my opinion, as somewhat of a biophysicist as well, I'm a little biased, but I don't think you can understand physiology in the human body without a solid understanding of physics. Okay, you've got to have that material in order to have a good understanding. So this is our goal for tonight, is to try to explain a little bit of the anatomy and the physics behind it and why it hopefully makes some sense. So we're going to cover only 3 different out of the 50 different functions in the body. So the first thing that we're going to do is we're going to talk a little bit about cardiovascular function. So some of you have some background on this, some of you don't. It's okay. We'll explain it along the way. Also, if at any point in time I'm going a little too quick, somebody raise their hand and get me to slow down. Okay? It's going to get a lot worse. So generally speaking, we're going to talk a little bit about how the heart works, some general anatomy and physiology. We're going to talk about some of the application of physics with heart function, and then applications to pathology with a couple of demonstrations. We're also going to talk a little bit about blood pressure, okay, the general anatomy and physiology of blood vessels, how the body pumps blood, and then the application of physics and some demonstrations with that as well. So I'm going to try to keep this relatively brief, okay, but when you are looking at the heart, we need to know some of the basic anatomy. Okay, first of all, your heart is roughly the size of your fist under normal physiological healthy conditions. It does lie within your thoracic cavity. This is about a 60-degree angle, and generally on the left-hand side, partially covered by your sternum, although some cases are on the right-hand side. Okay, so but that's generally where your heart lies, on the left side. when you break down the anatomy of the heart, you can do this in a number of different ways. But you're going to have primarily two upper chambers that are called atria, and you're going to have one on the right side and one on the left side. Okay, and I'm talking right and left side as the cadaver would lay. So not, as you're looking at me, but right side and then left side. You're going to have two lower chambers as well, and these are called ventricles. These are larger chambers. They're also more musculature. It's the job of these ventricles to take the blood from the heart and pump it to different parts of the body. So again, just the basic structure of this. The right side of the heart, you're going to see, is also not very muscular compared to the left side, and that's because the right side of the heart, its main job is to take blood that's already gone through your body. It's got very low oxygen, very high carbon dioxide, sending it to the lungs so you can get gas exchange. If you think about the distance that has to travel, it's a very short distance. So it's a lower pressure system. The left side of your heart is more muscular. What's going on? All right, and so that's responsibility of the left side of the heart is to pump the blood to the rest of the body, so it's got to be a bigger muscle to achieve that. It's a higher pressure system. Also with the heart anatomy, and this is starting to get into the physics part of things, valves separate the chambers and the blood vessels. So you're going to have basically two different types of valves. You're going to have valves that are between the atrium and the ventricles, cleverly named atrioventricular valves. On the right-hand side of the heart, you're going to have this thing called a tricuspid valve. Just means it has three cusps, three flaps. On the left side of the heart, you're going to have something called a bicuspid valve. This is the one that most people are aware of generally. It's called the mitral valve. So you might have heard about the mitral valve before, and that's the one on the left-hand side. Okay? The primary job of these vessels, or these valves, is to prevent blood from flowing backwards in the heart. Blood needs to go from the atria to the ventricles. We don't want it going the other way. That's pathology. We'll talk about that a little bit later. Also, you're going to have valves that are separating your ventricles from the blood vessels. So these are going to be called semilunar types of valves. On the right side of the heart, you're going to have something called the pulmonary artery. And the valve that's between the right ventricle and the pulmonary artery is known as the pulmonary valve. On the left side of the heart, going between the left ventricle and the aorta, you're going to have, again, the cleverly named aortic valve. So again, primary function here is to prevent blood, once it goes into a blood vessel, from going back into the ventricles. So again, we're chambering the blood and helping facilitate its flow through the heart. So how does blood actually flow? Blood is going to enter into the left atrium. I'm really going to focus just on the left side because that's where the physics is the most fun and where the pressures are the highest. Blood enters into the left atrium from the lungs, good oxygen, low carbon dioxide. It's going to pass through the mitral valve. It's going to enter the left ventricle. The ventricular muscles are going to contract. This will push blood through the aortic valve into the aorta, and then it's going to circulate the blood throughout the body. And I have a little demonstration, okay? So I hope you guys can see this, all right? Okay, so this is just a basic flow of where blood is going. Comes into the right side of the heart, right atrium, right ventricle, through the pulmonary valve into the lung, and it's going to come back to the left atrium to the left ventricle, through the aortic valve, back into the aorta, and throughout the rest of the body. You guys all right with that? Okay. Am I all right with that? Got that down? Little stumble, but not too bad. Thanks for not taking points off. All right. So let's talk a little physics. Okay, so how does physics help regulate this? This is about pressure and pressure gradients. So to understand this concept, we have to talk a little bit about diffusion. Okay, so diffusion is this universal law, and it applies to just about everything. Does anybody in the audience have a good functional definition of diffusion? Just anybody? Be brave. Fire away. Passive gas from one area to another, fine example. Okay, where does it move from? Does it move from an area of high pressure to low pressure or low pressure to high pressure? High pressure to low pressure. So this is the basic idea of diffusion. Okay? So things tend to want to move from an area of high pressure to an area of low pressure. Okay? Things want to move from an area of high concentration to low concentration. This is the basic tenet of blood flow. Could I get maybe 10 people up on the stage real quick? All right, everybody kind of gather, circulate in the middle part of the stage. Oh, look at that, nice and early. Tyler, get up here. You're not getting out of this. All right, fantastic. Get closer. 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 Make a circle. Get in close. I mean really tight. Closer. Closer. Now slide a little bit towards the screen. Excellent. Now get as close as you can. Push. You're uncomfortable now. All right, now here's what I want you to do. In your space, start to spin. Just start to spin. Keep spinning. Now put your elbows and put your arms like this. Put your elbows up. Start spinning. Keep spinning. Keep spinning and put your arms all the way out to the side. Faster. Faster. Stop. Diffusion. Okay? Thank you guys very much. Please return to the audience. Okay? Things want to move from an area of high concentration to low concentration. There is your demonstration. Okay? This is a law. This works for everything. It works for people. Okay? Have you ever been talking to people who get way too close? Okay, your immediate reaction is to back up. So everything that diffusion covers, okay, this is a regular thing. It happens in all systems. Works for pressure, works for volume as well. So let's do this. Valves are the key. Main job, remember, prevent blood from flowing backwards. Blood, as it enters into the left atrium, remember, we're just going to work on the left side of the heart. When it flows into the left atrium, blood is gathering, but it is not immediately passing into the left ventricle. The reason for this is, at this moment, the pressure in the left atrium with the blood is not as great as the pressure in the ventricle. So the pressure in the ventricle is slightly higher, which means as that blood is building, the mitral valve is closed. Does that make sense? As the blood continues to now passively return to the left atrium, you will see an increase in volume, but the size of the atrium stays the same. So when you're talking about an increasing amount of fluid in the same area, this will lead to an increase in pressure. At some point, that pressure in the atrium will become greater than the pressure in the ventricle. When that happens, it's pure physics. Physics doesn't think. Physics just happens. So when that pressure is greater in the atrium, the valve flows open, and the blood will now go from the atrium into the ventricles. This is a pure physics concept. Okay? So that's basically how that's going to go. Once that blood is now in the ventricle, the same principle will apply. Blood is now gathering in the ventricle, but it is not immediately passing to the aorta. Why? The pressure in the aorta is still greater than the pressure in the ventricle. So that aortic valve is closed. Now, as blood volume increases in the ventricle, okay, right, okay, so as blood continues to increase in the ventricle, at some point now your ventricle will start to contract. And as that ventricle contracts, it's going to increase the pressure in the ventricle. At some number, some pressure, the pressure in the ventricle is greater than the pressure in the atrium, and that will force the mitral valve closed. You can hear this. Okay, you can hear this. You guys know what that's called? Lub. Okay, it's your first heart sound. Okay, it's the sound of a door closing. Okay, so that's what you're hearing, and that's, again, physics. Now, for a fraction of a second here, and I mean a fraction of a second, the pressure in the ventricle, even though it's contracting, even though that mitral valve is closed, pressure is still greater in the ventricle than it is in the aorta. At some point, as that contraction happens, the pressure in the ventricle becomes greater than that of the aorta, and blood flows into the aorta. Pure physics. The valve opens. You guys all right with that? Okay, so again, pure physics. What prevents blood flowing back into the left atrium during contraction? Well, it's the mitral valve, isn't it? That nice tight sealing valve under normal physiological conditions. So when the pressure is greater in the ventricle than it is in the atrium, it keeps the valve closed. Okay? This prevents the blood from flowing backwards. So this is what a normal heartbeat sounds like. Lub-dub, lub-dub. Those are the valves closing. So under normal conditions, this sounds nice and clean. All right. So I'm just going to leave this here for now. It's going to make it easier. So let's do a little pathology. You guys have heard of heart murmurs before, right? Heart murmur is essentially an abnormal sounding heartbeat caused by the mitral valve not closing well. It's either got calcium deposits on it, there's some other type of issue with it, but it's no longer closing tightly. That nice clean sound you just heard, here's what it sounds like. Do you hear that whirring noise? What's the whirring noise? Blood. Can you hear the valves close? It's not that nice lub-dub anymore, is it? So with that mitral valve having those issues, that blood is going to flow back into the atrium. As it flows back, it's going to be turbulent sounding. And so you're going to hear that whir. Okay? That's bad. Okay? When blood is flowing from the ventricle back into the atrium, it causes a whole host of pathology problems. But the ultimate problem here is physics. Okay? The ultimate problem here is physics. If that valve is not tightly sealed, then those pressure gradients are artificial and they will not stand up to those pressures. Okay? And once the pressure now in the ventricle becomes greater than the pressure in the atrium, that valve closes, but it closes incompletely. So you're no longer getting good blood flow to your aorta, you're making your heart work harder, pathology ensues. You guys still okay with this? Any questions? You doing all right? All right, let's move forward. Okay, so let's talk a little bit about blood pressure. Okay, this is also physics. All this is physics. Okay, so what is considered normal blood pressure? Normally we'd say about 120 over 80. You guys have heard all this before, right? That's great. What does it mean? Okay, these are numbers. Okay, so what is that supposed to mean? Is it just a funky number? I don't know. Okay, this is describing systolic pressure and diastolic pressure. Systolic pressure is the high number. The systolic pressure is the pressure that needs to be overcome to open up the aortic valve. Okay? So generally speaking, that's about 120 what we call millimeters of mercury, and that is considered healthy, although there are charts that make this widely different. Okay, the most recent chart that I saw put 120 as pre-hypertension. Okay, that was a different number 5 years ago. Okay? Diastolic pressure is the lower number, and this is the pressure in the aorta after the blood has been ejected. Often this is kind of the pressure that allows the aortic valve to close. This is kind of like the normal keep the artery open pressure. Some people call it a resting pressure or a relaxed pressure, but those are those two numbers, and they are measured in this thing called millimeters of mercury. How is this measured? This is measured with a really fun tool called a sphygmomanometer. Okay, I think I said that wrong, but that's all right. Okay, it's a real word. You can use it in Scrabble and Hangman, no one will get it. Okay, so big points for that. Basically what this is you take a cuff, it inflates the pressure in the artery so that you close the artery, then you've got the stethoscope that you're listening to, and you put this over the artery and you're listening for these things called carot cough sounds. Okay? Carot cough sounds, the first one that you hear is going to be the systolic pressure, and when the sound stops, you're going to hear diastolic pressure. Okay? So what are we listening to here? Okay? We're listening to blood flow. When blood is flowing smoothly, it's called laminar flow, and laminar flow has no sound. It's completely smooth. When you compress the blood vessel and stop blood from flowing, the moment that blood valve, or that blood vessel opens just a tiny bit, it allows blood to squirt through. Think about like a garden hose in your backyard. That's turbulent flow. And so that blood creates turbulence as it's going through, creates little eddies that are in the fluid, and it bounces against the artery walls, and now you're hearing your noises. Okay, so these things are called carot cough sounds. When the sounds stop, that means the blood vessel is open enough that blood can flow smoothly, laminar flow. That'll be your diastolic pressure. So the first noise you hear, systolic. Second noise you hear, diastolic. Okay, this is not lub-dub. Okay, this is blood pressure. It's a different concept. Dr. Mahasma.
Thank you, Dr. Lewis. Well, you have to excuse my voice. Something is wrong with my voice today, so you have to excuse me. Let's look at a different side of this story. And you'll see why we're looking at the different side of it. Suppose that you're lying down on the floor and somebody puts a block, like a concrete block on your chest. Well, you feel pressure, right? Why do you feel pressure? Why should you feel pressure? If that was a styrofoam block, you would not feel that much pressure, right? Well, because the pressure is coming from the weight of that block that is on you. Okay, we all agree. Now, suppose that you're underwater. Let's say you're scuba diving and you're underwater. Should you feel pressure? Why? Because of the column of water, which is on top of you and pushing you down, which has weight, just like the block. Every cubic foot of water has a weight of about 62 pounds. Or 1 cubic meter of water has a mass of 1 ton, 1 metric ton, 1,000 kilograms. So that weight is pushing you down the same way that block of concrete or whatever it was pushing you down. So you feel pressure. Now, think about when you are just standing in air, no water or anything. Well, you have heard of the pressure of atmosphere, right? Where is the pressure of atmosphere coming from? Well, you have this column of air on top of you that extends for miles and it goes up. Granted, it gets thinner and thinner as you go up. However, at the sea level, on a calm day, the density of air is 1.29 kilogram per cubic meter, which means that 1 cubic foot of air has a mass or a weight of roughly about 0.1 pound. So air is light, but not weightless. So as a result of that, and because this column of air extends way out there, gets thinner and thinner, but it gets way out there, it does exert a pressure on you the same way that that concrete block or water column and so on were exerted on you. Okay, now the question is, how much is 1 atmosphere pressure? Well, we have all heard of it and we all talk about it. Well, here are just numbers. Probably don't mean much to you. 1 atmosphere is about 10 to the 5th Pascal or newtons per square meter, some SI unit, International System of Unit. In a more familiar system of unit, it's 14.7 psi. And it's also 760 millimeter of mercury. Where did that come from? Well, you have heard of barometers that used to be made with mercury, but nowadays they're not doing it because mercury is highly, highly toxic. On a calm day at the sea level, the height of the mercury in a mercury barometer is 760 millimeters. Then if the air pressure drops, column of mercury, the level goes down. If it increases, it goes up, and so on. So because on a calm day, pressure of atmosphere at the sea level is 1 atmosphere, therefore they associate this number, 760 millimeters of mercury, with what? With 1 atmosphere pressure. Okay. But now, the question is this. How big is 1 atmosphere? We are all living under 1 atmosphere, right? How big is it? Well, to give you an idea, suppose that there is an object with a mass of 2,117 pounds, and it's sitting on the floor over an area of 1 square foot. The pressure that this 2,117 pound object exerts to the floor is about 1 atmosphere. Now imagine how much force is that. Imagine being there, lying down there, and then that block being on top of you. That means that then you are under 2 atmospheres of pressure. And imagine what can happen to you. You're going to be smashed, right? Okay, so 1 atmosphere pressure is not a small pressure. It's a very, very large pressure. In fact, I wish we had this presentation in one of our big lecture halls that we could do some demonstration, and we could crush cans, like soda cans for you, so that you could see what 1 atmosphere pressure can do. If you evacuate the air inside a soda can and expose that to the pressure of atmosphere, the can is going to be crushed. Okay, well, no big deal. You can crush it by hand, right? But that happens in only 36 milliseconds. In only 36 thousandths of a second, the can is going to be completely crushed. Can you crush can? Sure. Can you do it in 36 milliseconds? Absolutely not. You would not be able to do that. Okay? So that is what 1 atmosphere can do. Now, let's go back to the person underwater. Now, I'm sure you agree that the deeper you go in the water, the higher is going to be the pressure. Why? Because simply the height of that column of water is going to increase. Okay. Now, Dr. Lewis mentioned about blood pressure. And he mentioned that 120 over 80, those are systolic and diastolic pressures in millimeters of mercury. But what I should tell you here is that these are gauge pressures. The same way that when you measure your tire pressure, which is a gauge pressure, when you measure your tire pressure and you say that the pressure is 32 psi, is that the absolute pressure inside your tire? No. That is the pressure that inside the tire is in addition to the outside pressure of 1 atmosphere, which was 14.7 psi. So these are called gauge pressure. So when they say that your systolic pressure, when you say that your systolic pressure is 120, Okay. That means those are in millimeters of mercury. Okay, and remember that 760 millimeters of mercury corresponds to what? To 1 atmosphere. Okay, so when you say that your systolic pressure is 120, it means that the pressure, the systolic pressure of your blood is 120 millimeter higher than what? Than the outside pressure. And your diastolic pressure when heart relaxes is 80 millimeters of mercury higher than what the outside pressure. Which means that as your heart pumps, going through systolic, diastolic cycles, consistently your blood pressure is between 10 and 15% higher than what? Than the atmospheric pressure. And that's why your arteries don't collapse because of the atmospheric pressure outside. Okay? But because those are gauge pressures, those are relative to the outside, that makes the absolute pressure in your heart at the systolic and diastolic stages 880 over what? 840. So these are the absolute pressures in your blood. Let's go back to this figure, this picture here. We have the same guy who was underwater now came out of the water and dried himself up, now is standing up. Now, here, oops, I keep pushing the wrong button here. Okay. Here is the pressure at the heart level, which fluctuates between, let's say, if it's an excellent blood pressure, between 120 over 80, it's just fluctuating in that level. That's the heart level. Now, because inside of the body there are veins and arteries and everything, there is blood there, right? Well, and blood is just a liquid, just like water, except that it's a little bit denser than water. Water has a density of 1,000 kilogram per cubic meter. Blood is 1,025. So it's a little bit denser than water. But the same laws of physics apply to blood that applies to water, which means that the lower that you are in blood, just like the lower that you are in a lake, the higher is what? Is the pressure. Therefore, because here is lower, what happens is that the pressure in your feet, or like at your ankle, is going to be higher than what? Than the pressure at your heart level. And the pressure in your head is going to be what? Lower. Just like being lower or high in a lake at the depth of water. Okay, now the question is, how much higher is your blood pressure here compared to your heart? Well, I just brought a meter stick here, and just before the presentation started, you can just measure the height or the distance between your, here, let me just throw it here. If you measure the distance between your ankle and your heart level, you will see that it's roughly around 1 meter, give or take. Okay. There's one other thing I was going to tell you earlier. Down here, I don't know if you paid attention or not. When you are deep, when you go to a depth of water, for every 10 meters of water, you get 1 atmosphere of pressure that is added to the pressure. So if you are at the surface of water, the surface of a lake, all you have is just 1 atmosphere pressure coming from the air. If you go down 10 meters, you get 1 extra atmospheric pressure, so the total pressure would be 2 atmospheres. If you go to 20 meters of water, 2 atmospheres comes from water, 1 from air, so you get 3 atmospheres total. Okay, fast forward. So, your ankle or your feet are about 1 meter below your heart level, right? Okay. Remember, for 10 meters of water, you get what? You get 1 atmosphere. Now you are down in blood a depth of what? 1 meter. Density of blood is very close to water, 1,025. Water is what? 1,000, very close. So it means that down here, you should be, how much higher pressure here? 1/10 of an atmosphere, exactly. And remember, atmosphere was 760 millimeter of mercury. So this means that your blood pressure down here should be 76 millimeter of mercury more. Therefore, if you add that to your 120 over 80, your blood pressure down here would be 200 over 160. So if you may take your blood pressure at your arm, which is the heart level, and if it reads 120 over 80, if at the same time you go and measure it at your ankle, you should get 200 over 160. Is it true? Well, time for demonstration, and we will find that out. We have two of my very good students here, Ben Lesko and Raleigh Bem, who are going to, Ben is going to measure Raleigh's blood pressure, both at the arm and then at the ankle, and see if this, what we said here was true. We'll find out.
125 over 85.
What I said was that if you're 10 meters underwater, the total pressure on you would be 11 atmospheres. One coming from the air and 10 atmospheres from water if you are at 100 meters underwater, right? So if you're at 100 meters underwater, 10 atmospheres coming from water and one on the top of water, so that's 11. So your blood pressure, you automatically adjust to being about 10 to 15% higher than the outside pressure, which is 11 atmosphere at the systolic and diastolic. However, you don't want to go too deep without proper suit, because if the pressure is too high, then other problems can develop, because your body may not be able to handle that much pressure. Your heart may not be able to handle that much pressure. So what did we have?
210 over 162. 210
Okay.
over 162.
So what did initially? 125 over 80, and then we got 210 over 162. So as you see, pressure at the ankle is considerably higher because of the variation of pressure with depth. Okay, I think. All right. Well, we're going to look at sound and what we perceive as sound and what we perceive as color and image in general. Well, we're going to start with a brief discussion of waves. We have all seen waves, right? At least we have seen waves on the surface of water. But there are generally two types of waves. One type that is called transverse wave, a wave that moves like this. Okay, that's one type of wave. And another type of wave that moves like this. Okay. The first type of wave is called transverse wave. The second type is called longitudinal. And which wave you have in a given situation? Well, it depends on how the wave is generated. What you need to know is that anytime you have something oscillating, anything oscillating, it generates some sort of wave. And conversely, anytime you see a wave, there is a source for that wave, which is an oscillator. Sometimes that oscillator may not be very trivial, might be very complex. However, there's always an oscillator that generates a wave. Now, let me show you some picture of these waves. The first one up there is transverse, and down here is longitudinal. And that is just shows the wavelength in each case. That you see that you have wavelength there, and in the longitudinal wave also you have a wavelength here. Now, let us suppose that you take a fishing bobber, you put it on water, and you attach a line to it, and it just oscillates it up and down gently. Well, that generates a wave, as you know, right? Let's call that source. That is the oscillator that generates this wave on the water. And let's say you put another bobber here, a free bobber, and let's call it detector. As the wave reaches this bobber here, this one is going to respond and is going to start oscillating, right? Now, no matter what happens between here and here, the frequency of oscillations of this bobber is going to be the same as the frequency of the oscillation of that bobber. In other words, the frequency of the detector is always going to be the same as the frequency of the source, no matter what. Wavelength can change, the speed of wave can change, many things can happen, but frequency does not change. So that bobber is going to mimic the frequency of what? Of the source. Okay, now, suppose that I'm going to skip using the speaker to save time here. Suppose that I take a piece of paper and then do this. What happens here? Every time I move this forward, it pushes air towards you. When I pull it back, it pulls it back. And this is an oscillator. And as I said, every oscillator generates a wave. What kind of wave is this? This is a longitudinal, exactly, a longitudinal wave. A longitudinal wave in pressure of air. It's a pressure wave that is longitudinal, and it keeps on coming like this towards you. Okay? So this is the source, and that wave keeps coming towards you, right? Until it reaches your eardrum. Well, when it reaches your eardrum, then what happens? Let me show you something else before I put the next slide on. When it reaches your eardrum, just like that bobber that I showed you, is going to respond to that wave. So your eardrum is going to oscillate. And that oscillation is going to be transformed into an electrical signal that Dr. Lewis will explain to you. And that electric signal is transmitted to your brain. What is your brain going to do? Nothing. Doesn't realize it. It does not recognize anything. But suppose that I go faster and faster and faster and faster. Suppose that I can do this 20 times per second. Then your brain is going to say, aha, sound is coming. And if I increase that frequency, the pitch of the sound is going to go up. Okay? And eventually, let me just quickly turn this on. And okay, look at the drum of the speaker. You see that move going in and out? You can't see it from back there, but those who are sitting closer, you can see it's going in and out. See, that is generating that pressure wave, longitudinal wave, and I'm increasing the frequency. You still can't hear anything. But if I increase the frequency, now it's 31 times per second. Now you can hear something. And if I keep increasing the frequency, the pitch keeps going up. Then come and feel the drum of the speaker. You see that it's vibrating. Right? And then as you increase the frequency, see right now the drum of the speaker is oscillating 452 times per second. Okay? So now your brain is recognizing that. It's the same type of wave that was reaching your brain when the oscillation was small, but your brain did not recognize that as anything. But now that frequency is high, the brain recognizes it. So from about 20 oscillations per second, your brain recognizes that as wave, as sound, up to about 20,000. But that upper limit drops very quickly with age. I cannot hear even 16,000. That tells you how old I am. Okay. So anyway, so going back to what I was just saying, the drum of the speaker is nothing but just a plane that is moving back and forth and generating those longitudinal waves that are pressure waves in air. They reach your ear, and they enter your ear, and your eardrum responds to that exactly the same way that the bubble on the surface of water was responding. And then that oscillation is converted into an electric signal, which is transmitted to the brain, and Dr. Lewis is going to explain to you what is going to happen inside.
Thank you, Dr.
Thank you.
All right, so my turn. Let's do some of the physiology part of this. So when we're looking at wave function, longitudinal waves, we already have established now that the waves that are created are pressure waves, and they're going to have an effect on the ear that theoretically are going to lead to a sound that your brain can recognize. So I'm going to do this relatively quickly. Sound waves that are generated are going to hit the thing called the tympanic membrane. Tympanic membrane is now going to oscillate at the same frequency that Dr. Mahasby was talking about. This tympanic membrane is going to be resonating with bones of the middle ear. You know about these, the malleus, naecus, stapes. And this vibration of the tympanic membrane is going to be transmitted to something called the oval window. There's another membrane. This is going to be on your inner ear. Okay, so we have the outer ear, the bones are in the middle ear, and then we're going to go into the inner ear. When this oval window vibrates, it will still be at the same frequency. Nothing has changed. That frequency, that vibration, is now going to cause waves within fluid that exists within your inner ear that is known as paralymph or endolymph. We're going to focus on paralymph. When this fluid now is oscillating back and forth from this membrane, it's going to cause the displacement of a series of cells that are known as the basilar membrane. These will actually start to shift. When that happens, it's going to displace these structures here that are known as hair cells. Okay? Now, they're in your ear, okay? And these are not hair follicles. These are modified neuronal tissue. They're called hair cells. And this is what they look like with an electron micrograph. They look really cool. Okay? All right, so these hair cells now, as the basilar membrane is causing them to oscillate, these hair cells are going to go back and forth. You with me on this? Okay. Now, as they go back and forth, it's going to cause these membranes, these hair cells, to expand and contract. When they expand, it's going to open up a really cool molecule known as a stretch-activated potassium channel. Okay? There's a whole other lecture on that. Take neuro. It's a great class. Okay? So the stretch-activated potassium channel is going to open up, and this positive potassium is going to come into the cell. It's going to cause a depolarization. For those of you who had my neuro class, yes, I know that's opposite. It's okay. Okay? Specialized cell function. This potassium, when it comes into the cell, is going to cause a depolarization. The depolarization is going to allow for a chemical called calcium to come into the cell. Calcium will then trigger the release of these things called neurotransmitters, these chemicals, which will now stimulate your nerves that will create an electric signal, again, physics, another lecture. That electric signal called an action potential will now go to your brain, and your brain now has the opportunity to perceive this as sound. Here's the pathway for anybody who cares. Okay? Hair cells, vestibulocochlear nerve. Yes, that's number 8, and yes, you should know that. Medulla oblongata, inferior colliculi, thalamus, auditory complex of the temporal lobe and the cerebral cortex. Neuro gets the fun words. Okay? So when it gets, and by the way, that's right about here. All right, so, and that is where sound is going to be processed. Your brain is now going to look at and listen to nothing. There's no sound. This is a binary code. It's ticks on a screen. Okay, so it's going to be listening for the frequency of action potentials. Pitch, as Dr. Mahasby was pointing out, is based on which hair cells are stimulated within this conch shell structure known as the cochlea. The hair cells that are closest to the oval window are the highest pitch. As you move around this cochlea, you get to lower and lower pitches. Okay, so that's where you get the frequency from. Also, really cool, in your brain, in this temporal lobe region, you actually have a map that's on there that has been hardwired through your life so that when that electric signal, that action potential goes to a particular part of your brain, you are now interpreting it as a high sound or interpreting it as a low sound. It's called a tonotopic map, okay? Similar to something called a somatotopic map. Now, volume, loudness, decibel level. Hair cell movement causes these action potentials. What your brain recognizes as volume is the frequency. So if I'm talking relatively quietly, I am not creating very large amplitudes. Okay, so this is only causing a small vibration of your tympanic membrane that's causing the hair cells to move a little bit, which causes just a small increase in action potential frequency that goes to your brain that says this is not a very loud sound. When I start talking like this, I'm creating a fairly large amplitude, which is going to severely move your tympanic membrane. You with me on this? Okay. That's going to cause major displacement, right? And that's going to create a larger frequency of action potentials. So your brain recognizes volume by the number of action potentials that reach your brain per unit time, also frequency, also physics. You guys good? Okay. Hearing loss, just a really quick diagram of this. It's a little bit hard to see. Do you see how nice and pretty this looks? That's normal healthy. Inner hair cells, outer hair cells, another lecture. This is bad. Okay? This is age. This is damage. So those hair cells start to deteriorate over time. Before anybody asks the question, we're not really sure exactly how this happens yet. Leading candidates are what are called normal old aging. Increasing free radicals as you age cause disruption of plasma membranes, which lead to deterioration of cells. So, all right. Dr. Mahosby, really quick. I don't think we have a huge amount of time. You're up.
Thank you.
You're welcome.
Can you hear me? Okay, good. Okay. Suppose that you pick up an object that has some electric charge on it. That's easy to do. You comb your hair. I don't have hair, so I can't do that. If you have hair, just comb your hair. The comb would pick up some electric charge, as you know, right? Okay. So suppose that this is a comb, and suppose that I had hair, and I comb my hair, so that has electric charge. Then suppose that I start oscillating it. Well, remember I told you anything that oscillates generates some sort of wave. Here you're oscillating electric charges. What kind of wave does it generate? It generates a transverse wave that is called electromagnetic wave. And the reason that it's called electromagnetic wave because the wave that comes towards you has two fields. One is electric field that oscillates like this, and the other one, magnetic field, oscillates perpendicular to that, and the wave comes towards you. Excuse me. Now, suppose that you have a radio, and if you had a radio here, it would work. What happens is that this wave that is generated reaches the antenna of your radio. In the antenna of your radio, which is a metal, excuse me, you have electrons and protons in the nuclei of the atoms. The electrons would respond, would oscillate the same way that that bubble that I told you on the surface of water would respond to the waves. The electrons would oscillate here, responding to the wave, and that oscillation is transmitted to the radio, and the amplifier amplifies that and sends it to the speaker, and you can hear it. So if you had a radio here, you could hear click, tick, 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 tick. And if I go faster, tick, 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 you could hear it. In fact, in Green Course 101 that I teach, there are speakers there that every time I do this, students can actually hear it on the speakers. So what happens then? If this was a charged object and I was doing this, the wave would come towards you and it would enter your eye. Okay? So then what happens? Then the wave enters your eye and it reaches the optic nerve of your eye. The optic nerve of your eye is a conductor. It's not metal, but it's conductor. Just like the antenna of the radio. So what happens is that the electrons in that optic nerve, they start responding to that electromagnetic wave the same way that the electrons in the antenna is responded, or the same way that the bubble on water responded. So the electrons would respond, would oscillate, and they generate an action potential that Dr. Lewis mentioned, and that signal is transmitted to your brain. What is your brain going to do with it? Nothing. Doesn't even recognize it. But suppose that I go faster and faster and faster. Okay? Suppose that I was a Superman, and suppose that I could oscillate this 430,000 billion times per second. Okay? Suppose that I could oscillate this with a frequency of 430,000 billion times per second. Then you would see red light coming from here. Okay? And if I go faster and faster, the red light would gradually change to orange, yellow, green, and eventually at about 790,000 billion times per second, the light becomes blue, and if I go faster than that, the light disappears. Why? Because then it would be emitting UV, ultraviolet electromagnetic wave. And then if I go even faster than that, it starts generating or emitting X-rays. And if I reach about 10 to the 18 times per second, then you will all be running for your life because I would be generating gamma rays. and then a few hours after that, I'll be dead too. Okay? So anyway, so your brain would recognize those oscillations if the frequency of oscillation, and again remember, the frequency with which those electrons in the optic nerve oscillate and send signal to your brain It's the same as what? As the frequency of the source. And if that frequency is between 430 to 790 terahertz, which is the number that I gave you, then you would see light. This end is red. That's why I wrote it in red. And this end is the blue end of the visible spectrum. Dr. Lewis, your turn.
I can do this. All right. Thank you, Dr. Mohosby.
You're welcome.
Now, for tonight, we're not going to worry about color because we're just not going to have time. Okay, so what I want to talk about is black and white a little bit. So in your retina, you've got these different types of photoreceptor cells, things that are called rods and cones. Rods are for black and white. Cones are for color. Within these photoreceptor cells, you're actually going to have the ability to absorb that energy that Dr. Mohosby was talking about. These will be absorbed by proteins. Okay, and these proteins are going to absorb that energy, and they will create biochemical pathways in the cells in your eye, in your retina, that will be translated into electric signals that the brain can recognize. These are rods because they look like rods, and these are cones because they're cone-shaped. All right, so in the rods, the protein that we're talking about here that's going to absorb energy is something called rhodopsin. Rhodopsin has two components, this thing called retinal, which is derived from vitamin K. What did your parents tell you to eat when you were young to improve your vision? What are carrots rich in? Your parents are smart. All right, I say that as a parent. When the light is absorbed by this protein, it becomes unstable. When it becomes unstable, rhodopsin splits into two components. It's going to be this thing called opsin and this other thing called retinal. That will now create an action potential that will follow this pathway, optic nerve thalamus, optic chiasm. You guys know that things cross over. Okay, this happens within the visual pathway. It's going to go to the striate cortex, the occipital lobe in the cortex. Again, we get the best names. And this is also called your primary visual cortex or the line of generic for anybody who's a historian. Okay, so that's where you're going to start to process that information. Let's talk about two things really quickly to kind of end up. Night vision. Okay, can you see, well here, let's do it this way. If you're in a light room and you go into almost a completely dark room, there's still a light source, but it's very faint. How well can you see when you go into that room? Initially, not very well, correct? Okay. But now after you're in there for a couple of minutes, you can see better, correct? Why? Your eyes have adjusted. Best answer ever. Okay, but what does it mean? Okay, you're right. So what? Okay, so when your eyes are adjusting to that, what does that mean? Well, remember that light breaks up the photoreceptors. So if you're in a dark space, those two proteins, opsin and retinal, will come back together. They will form this functional rhodopsin, and the main function of rhodopsin is to see light. And we're talking about a white light source here, so black, white, very binary. So the longer you're in the dark, the better you're able to see. And it has to do with the percentage or the concentration of those intact proteins within your photoreceptor cells. That is night vision. That is how it works. Okay, that's called dark adaptation. Here's the opposite of that. Ever been in a really dark room and someone turns on a light? What does that do to your eyes? What do you want to do? It's too bright. It's not too bright. Why are you more sensitive to that light? Because you are just in the dark, and you have a higher concentration of those proteins intact. So you now become more sensitive to the light that is there. Does that make sense? It's called the bleaching effect. Stargazing, one of my favorite activities. Okay? When you are stargazing, if you've ever noticed, if you try to look directly at a point source star, you will lose it. You will not be able to see it as well. The reason for that is when you are focusing on something directly, it goes onto a part of your retina called the fovea centralis, and the fovea centralis is full of cones. There's no rods. Cones are for color, right? There's no rods. So if you're looking directly at the source, you have no way to see it. Look 1 degree to the left, 1 degree to the right, 1 up, 1 down, pop right back into focus. So when you're stargazing, try not to look directly at it. You'll see it more clearly if you look to the side. This also works for meteor showers. Keep an open gaze. Don't try to focus too much on one thing. You will often see it in your periphery. Trust it. It's happening. You're picking up faint light sources because you're not looking directly at them. So again, as you're out stargazing or meteor showering, and by the way, the Lyrids are coming on April 22nd. It's supposed to be a new moon, so it should be really good to go out and look at meteor showers. So, but that's basically how some of these things work. All right. I think we're done. Thank you all so much for coming. Our assistants need to come up here as well really quick.